Okay, so now we're moving on to section 9 of chapter 3, where we're going to talk about the nucleus, which is pretty much the control center within the entire cell, and it's a place where we contain our genes or our genetic information. I have on here study table 3.4. 3.4 is a really good table to kind of summarize all of the different organelles that we've talked about so far, and their function as well as their location, including the plasma membrane, our cellular extensions, or even our microfilaments and centrioles and things like that. So the last structure inside the cell that we needed to talk about, again, is the nucleus. When we talk about the nucleus, we talk about it as a structure as a whole, but it's actually made up of 3 different things. So we have our nuclear envelope, our nucleoli, and our chromatin. We probably think, because it contains our genetic information, that every single cell in our body contains at least one nucleus. However, that's not the case. One of our cells, our erythrocytes or red blood cells, actually does not have a nucleus at all. So we refer to erythrocytes as being anucleate. Most of our cells do have one nucleus, so we say that they're uninucleate. And then we have a couple cells in our body that have multiple nuclei, and we say they're multiple nucleate. So when we hit the skeletal muscle system, you'll see how a skeletal muscle cell actually has multiple nuclei in them. So let's first talk about the structure of the nuclear envelope. It's a double membrane, just like we saw with the mitochondria. It's going to enclose what we call the nucleoplasm, because this is our fluid within the nucleus. So this is distinct from the cytoplasm. The outer layer of the double membrane is continuous with the rough endoplasmic reticulum, which is why the nuclear envelope is a component of that endomembrane system. It also has a nuclear lamina, and this is just a network with a mesh of proteins that's going to act to help maintain the shape and acts as a scaffolding for DNA. So this component is similar to our cytoskeleton that we had in our cytoplasm itself. Another important feature of the nuclear envelope are nuclear pores. So all these perforations within the nuclear envelope are really important so that we can transport and import different things to and from the nucleus. Within the nucleus itself, typically at its center, you have at least one nucleoli, and the function of the nucleoli is to synthesize ribosomal RNA or rRNA. And if you remember when we were talking about ribosomes, we said that ribosomes are made up of rRNA, and rRNA is not an amino acid, so it's not a protein. So all the structural components needed to make our ribosomes are actually produced within the nucleus, and more specifically within the nucleolus. Typically a cell has 1 or 2, but I think in lab your structure of the nucleus is actually going to show 2 nucleoli. And of course, we have to have our genetic information, which ours is in the form of DNA. So we have something called chromatin. Chromatin is what's going to make up our chromosomes, and typically we might think that chromatin is solely DNA. However, there's other structures in there as well, such as proteins called histones, and we even have some RNA in there as well. We arrange chromatin into these fundamental repeating units that we call nucleosomes, and then we can go in and alter the histones by attaching some sort of tag like a methyl group or an acetyl group in order to regulate gene expression. Because within every nucleus that we have the same genetic information. So how do we have a liver cell that looks like a liver cell and functions like a liver cell that has the same DNA as, say, a cardiac muscle cell that functions in our heart? They both have the same DNA. How are they structurally and functionally different? And that's by altering the amount of DNA that is available for gene expression. So we always have chemical tags on our DNA, specifically the histones, so that we silence some genes or we allow expression of other genes. So here's just a visual. To your right over here, this is going to be our DNA double helix, which we'll talk more about in a little bit. These are those little proteins called histones. They come together, 8 of them will come together, and when 8 of these histone proteins come together, they're going to provide the structure around which DNA can wrap about 2 times. Once we have our DNA wrapped around these 8 histones about twice, we refer to it as a nucleosome. So this is one nucleosome, this is another nucleosome, and so on and so forth. And again, these are our structural units of our chromatin. If we take this structure where we have our DNA wrapped around these histones and we look at it under an electron microscope, it's going to look like this. So this model is also referred to as beads on a string because it literally looks like we have some beads on a string. So once we have our DNA wrapped around our histones, the next way that we further compact it down is to basically turn this beads on a string into a helical fiber that kind of looks like this. Then we're going to compact it down a little bit further where we get chromatid. And then we're going to take our chromatid and wrap it around a scaffolding and form what looks like the chromosome right here that's shaped like an X that we're used to visualizing or seeing illustrated when we talk about chromosomes.